I'm pleased to introduce now our next speaker. Mehrdad Yazdani is from Biome and UC San Diego. Mehrdad's favorite part about being a data scientist is being in everyone's backyard. He works with domain experts ranging from social scientists and artists to biologists and clinicians. In addition to applied work, he also continues to do research primarily at UC San Diego and teaches courses on topics related to machine learning, data mining, and neural networks. He will be presenting a talk titled Machine Learning in Healthcare. Please join me in welcoming Mehrdad.
Thanks for the introduction, and thanks for coming back after lunch. So I'm going to ask a question that I think is appropriate for this audience, or for this conference. How many people here think big data is a buzzword? And the question is, so buzzword's kind of a negative connotation, right? It's kind of like this trendy thing, and maybe it had seen its peak, and its height is gone, and it's not that cool anymore. And I think maybe in certain verticals, if you will, that's certainly true. But I think there are still lots of potential for big data technologies and infrastructures to make huge impact. I think one area is healthcare. And a lot of my work is inspired by this guy, my boss at UC San Diego, Dr. Larry Samar. He's the director of the Kalatizhu Institute at UC San Diego and UC Irvine. And right around 2000, he decided that for various reasons, he wanted to quantify as much as he can of his body. Right? So he's kind of a believer in the quantified self. And really what his main motivation was his own health. He wanted to make sure he's healthy, and he wanted to collect as much data as possible. He's an astrophysicist by training, so he believes in big data, right? So around 2000, he had one data point. He was just looking at his weight, right? Like every day, he was looking at his weight. And then eventually, he started going to the lab more often, getting different lab values, and kind of on a regular basis, getting these various blood biomarker data points. Then he started sequencing his genome, which gives him orders of magnitude more data. And now he's regularly sequencing his microbiome, his metabolomics, his proteins, proteomics, and so he's at many billions of data points. And this is helping him decide how to change his lifestyle, change his diet, change his treatment for his diseases. He does have some illnesses, and he's using data to guide him in how to treat himself. Now imagine this. He's the director of a lab. He's got lots of people working for him. Imagine this capability bringing to the general population. Right? So imagine this scale of data for now 1 million people. And maybe today people are not interested in this, but you can imagine by the law of exponentials that almost surely 1 million people will be very, very interested in knowing about their own body. Right? We're used to going to the car automaker to get our car and oil change, and the mechanic will run a bunch of different numbers. Why not our own body, right? So this has really motivated me to look at these types of data sets and these biological data sets. And I want to show you this. The central problem here is, this shouldn't be happening. But so the central problem is that we don't have just a growth in the number of patients. Right, so if you start a website, a social network website or something, typically the big data problem you have is growth in the number of users, right? Like you have millions of people joining, hundreds of millions of people joining, and then that's like a huge growth in the number of users, right? But in biology, what we're seeing is that we also have an exponential growth in the number of biological measurements. And these biological measurements, as the science of genomics and different omics is improving, the number of different kind of biological features that we're interested in is continually growing. And so the data science problem here is how do you deal, not only how to deal with a growing number of patients, but also a growing number of features. So for today, I'm just going to talk about at a high level of one particular type of biological data, and that's the microbiome. So I'll give you the overview of the microbiome. And then I'll show you some popular machine learning methods that are used into this particular area of biology. And then I'll give you a sample work that we did at UC San Diego, trying to identify the major protein shifts in the gut microbiome for irritable bowel disease. So let me start with the microbiome. So So this is the tree of life. You may have heard of the tree of life. And this is basically the tree of life is supposed to kind of categorize and organize all the different living organisms into this kind of hierarchical structure, right? And so there's three major types of life forms. There's archaea, bacteria, and eukaryotes. And practically any living thing that you see is that little green sliver. The overwhelming majority of living things on this planet are bacteria. This kind of shaded area it's called candidate phyla. That's just, we're not quite sure exactly how it fits, but these are all basically bacteria. So the overwhelming majority of life forms on this planet are bacteria, and they're really running the show here. And you know, we're of course we're much bigger than they are in terms of size, but in terms of numbers, they're huge. And so you might wonder, well, why do we care about this bacteria? Like, why is it so important? Well, the story for that actually is really interesting. So every single person in this room, every single person you know, in life started as one cell, right? And then that one cell divided into two. And then the two went to 4, 8, and so on. And what's really remarkable is that as that process is happening, this exponential growth of cells, of you, is occurring, those cells divide into skin cells, some of them divide into hair cells, neurons, blood cells, right? So it's really remarkable. And so they keep growing to form you, right? And while this is happening, as you're growing, other cells start joining, namely bacteria. They start joining this party, right? And by the time you're an adult, they outnumber you 10 to 1. So 90% of the cells that are in your body are actually not human. They're bacteria. And so people who are studying, if you're interested in your own health, you should also be interested in the health of the microbes that are living in you. Because you live in an ecosystem with this collection of organisms that outnumber you 10 to 1. So here's a sample study. These different colors kind of show you the different types of bacteria that are in this one subject. And this subject was collecting different types of bugs, different types of microbes that's living in him over the course of a year. And you can see that it's relatively stable until the person goes and lives abroad. I think he was in Thailand for a month or so. Right? You can see there's this major shift. The green proteobacteria starts suddenly dominating, right? And this is a healthy person, but just the mere change in diet had a major shift in his microbiome. So how do we study the microbiome? It's not like studying animals where you can just go outside and observe and see how the cheetah is running around, or the elephants are running around and stuff. You have to actually go dig into the DNA. And so the reduction of cost of sequencing technology has really made this an opportunity for us to study all these different microbes. And this is what makes it a heavily intense data problem, right? Because what happens is we take the DNA and we get this sequence of genomes out. So the diversity that we have is actually quite wide. You can imagine that if you go to a desert like this, you'll have a completely different experience than exploring this coral reef, right? You'll run into very different animals and species here versus the coral reef, right? Just completely different. Now the microbiome in our body, the difference between this desert and the coral reef, that huge difference is roughly similar to the difference we have in our skin and our gut. Now, so there's microbes living in your skin, and that's one particular type of ecology, and there's another ecology in your gut that's also vastly different. And as you can imagine, just this very small distance between your skin on your hand, say, and your gut, it's a very small distance. It's as wide as the distance between the desert and the coral reef. But this makes it a very challenging problem, and this is why machine learning is increasingly becoming an important tool in understanding how are these ecosystems different and how are they similar. And really the main question is, how do we measure similarity between different types of ecosystems? So this is a very complicated process. It requires lots of scientists from different disciplines working together to analyze these different samples. So what happens is people usually take samples from different parts of the body. If it's from the gut, it's usually the stool. So perfect after lunch, right? I can talk about that stuff. And so they take the stool, they prepare it into the lab, they pass it through a sequencing machine, they get the A, T, C's, and G's, and then those A, T, C's, and G's are classified in a biological database of known species that are then organized into this phylogenetic tree. Right? And once we have one sample organized into this phylogenetic tree, then we can actually kind of see how those compositions are different from different body sites, and we can compare them in a PCA type plot. Okay? So this is a study that is looking at the PCA of the different microbes. Technically, it's the PCOA because they're using a different metric, but we won't go there. PCOA, also known as multidimensional scaling, or not. But those are just little details. So this is two subjects who live together, and there's samples from their gut, their hands, and their mouth, their tongue, right? So you can see that on the top there, this is the gut samples, and on kind of the horizontal axis is time, and these are the two top principal components. And as time goes by, the male and the female, their guts are pretty stable. They're kind of in this region of the PCA space. The tongue is also pretty stable, and then their hands are kind of also stable, but they have significantly more variance. And you can imagine the reason there's so much variance is because they're exposed to different things. Now, this male and female, they're actually a husband and wife. They live together, and that's why their tongue microbiome is so similar. But if you take two different, and their gut microbiome, right, because they share a similar diet. But if you take two random people, their gut microbiome and their tongue microbiome will probably be different. So in this study, we actually used a more sophisticated technique because we were not interested in just separating the different body sites, but we also wanted to separate the husband and wife. So we used this technique called topological data analysis, provided by the software provided by Yazdani, that we were able to actually not just distinguish between different body sites, between gut and skin and the tongue, but also the different subjects, right? The female and the male, right? So being able to untangle these patterns using more sophisticated machine learning, we're actually able to kind of figure out what are the differences between these subjects and body sites. So this type of machinery is not just kind of for science, but it's also very practical. And so I used to work at this company called the Open Medicine Institute, and we developed this pipeline where you would actually take actual patients, we would sequence their stools, we'd be able to tell them what is their different compositions of their microbiome, and then using a PCA, we'd be able to tell them where does this patient land into this PCA space, right? So these different colored dots indicate different cohorts, right? So we have the kind of the orange is the healthy cohorts, then we have different types of IBD, the colonic Crohn's, oops, sorry. Oh my gosh, there we go. So we have colonic Crohn's, and the patient in this case is kind of lands right here, and so they're somewhat on the fringe of healthy and colonic Crohn's. Right? And another company called the American Gut, they're actually a nonprofit at UC San Diego. So if you're interested in sequencing your own microbiome, you can send them a sample, and they'll tell you, where does your sample fall in terms of its kind of relative abundance of different microbes. And they'll also give you a PCA plot that shows you in their population, where does your sample fall compared to everybody else. So using these kind of PCA and dimensionality reduction techniques are really valuable for us to understand this huge variety of species and understanding how these ecosystems are different and similar. So in this particular study, we were really interested in figuring out what are the driving kind of forces behind these changes and shifts. So in this work, we looked at, this was work done at UC San Diego. We took, there was a microbiome project that provided us 33 healthy individuals, and we also had 28 samples from irritable bowel disease cohorts, and the cohort kind of had sub-cohorts of different types of irritable bowel disease. And what we did here was we actually sequenced the DNA into not just the different types of species, but the different types of proteins, right? So the number of species, bacterial species, it always changes, it's always growing, but it's typically on the order of thousands, maybe tens of thousands. But the proteins are on the order of millions. Right? They can be on the order of millions. But at the time of this study, our proteins were on the order of tens of thousands, and what we wanted to know was, is there a difference between doing a PCA using species versus proteins level, right? So this is kind of the same data. On the left, we have the species level classification, and on the right, we have the proteins level classification. And as you can see, actually, there is sort of the separability between the different cohorts is not so strong when you're looking at it at the species level, right? So if you're looking at just the species that are in your gut, they don't quite separate out so neatly the different cohorts. The green dots are the healthy cohorts, by the way. But then if you use the protein level, you can see that at protein level, the green cluster, the healthy cohort, they just kind of form their own island, right? So that's a pretty robust, it's a far more robust signal there than using it at the species level, right? So this tells us that the proteins are telling us a lot, right? Because so you can think of species as the what are the bacteria that are in our gut, and the proteins are actually telling us the functions. In other words, what are the function of these different microbes? Right? So the function is actually telling us far more information than just what is in there. Okay. So the problem that we have, and this is kind of something that we're preparing ourselves for, right? Is that we have, like I showed earlier, right? We have a set of, in this case, the columns correspond to patients, and the rows are the different proteins that are classified in our KEGG database. So this is a KEGG is just one particular database of proteins, and this is on the order of 10,000. And the idea is, can we actually predict if a particular KEGG, if one of these particular proteins, is over or underabundant compared to a healthy cohort, right? And what we really want to know is, what are those particular KEGGs, what are those particular proteins that are important in distinguishing between healthy and sick? So in our KEGG database currently has an order of 10,000 or so proteins, but we expect this to grow to millions. And so what we really wanted to do was build a machinery that would be able to scale from going from tens of thousands to million. So the way we did this was we kind of prepared this basic pipeline, and what we did was we kind of decided to have these sets of proteins classified into kind of two separate groups, right? We have a current existing group, and then the idea of future proteins. And so what we wanted to know is, can we build a classifier using just the current existing proteins to classify future KEGGs that will be added to the database, right? So in other words, what we wanted to know is, as more proteins are added to the database, would we be able to predict if these proteins should be good proteins or bad proteins of these people? Okay. And so this pipeline, that's kind of how we do this, right? So the way we do this is we take those 10,000 KEGGs that we have, we split them into this kind of 50% random training set and 50% random holdout set. And so this kind of training set is simulating our idea of having a known database, right? And so from this 50% kind of random training set, what we do is we identify the most discriminative, the 100 most discriminative KEGGs using a classic statistical technique called the Kolomogorv-Smirnov test. And those 100 kind of key candidate KEGGs, we use that to build a random forest classifier, which we then use to see if we can actually predict this held out proteins, if we can predict them to be good or bad KEGGs. So that's just an explanation of that. So if we actually look at the features of these KEGGs, and each dot here now is a protein. And the colors are the confidence score of our classifier. So you can see our classifier has actually been able to partition the PCA landscape of all these different proteins into kind of meaningful groups. Right? So if our classifier had been overfit, if it was an overfitting classifier, you would expect a random gunshot scatter plot of these different dots. Right? But you can see that there's like sort of the low scores are all clustered here, then the medium range is kind of like, in the gray, and so there's this kind of very nice or organized distribution of these KEGG scores in the PCA space. So kind of having developed this technique, and this is the fun part where you get to work with biologists and actually see, okay, this thing that I built, how is it actually telling us anything that's biologically meaningful, right? So this, what we were able to do is we picked out these particular KEGGs and verified in the literature with the help of a biologist. I'm not a biologist, so I'm not a real scientist, just a data scientist. And gave the real scientists to let them figure out if is there literature supporting this? Does it make sense? And there is certainly literature that supports this, so that's good. And so we have now this pipeline that can scale to kind of any new protein. We're able to classify if it's good or bad. Okay, so that's kind of brings me to the end of my talk. I just wanted to mention one more thing. So predicting the future is always fun. One trick is you just want to maybe look at the laws of exponentials. So for example, in computing power, right? If you look at Moore's law in the 70s, if you extrapolate Moore's law, you would be able to predict almost surely everyone's going to have a computer in their pocket, by the 21st century, right? And that's certainly what happened. I think if you look at today's exponential growth, that curve belongs to sequencing, or at least one of the places that you see it happening is sequencing, right? So this is the reduction in cost of sequencing DNA. And you can see that, in the early 2000s, it was on the order of millions of dollars. And now that has been reduced significantly to, on the order of the hundreds of dollars, right? So you can imagine that a few years from now, everyone's going to have a sequencing machine in their home. And the future that a lot of us are kind of seeing here, which is not that far from now, is once you have a sequencing machine in your home, you can essentially have a lab in your bathroom. So that every time you go to the bathroom, you have a sequencing machine that's able to sequence your discards and keep track on a regular basis. How is your microbiome changing, right? Is it changing based on maybe something you ate, or is there something more long-term happening? And so we would be able to actually keep track of your state of health on a regular basis. So imagine the big data volume we'll get there, right? Every time you go to the bathroom and flush, terabyte of data. Okay. I'd like to thank my collaborators, Larry Smar, Bren Taylor, Rob Knight, Justine Debelius, Wei-Zhong Li, and Andy Kovalnik. Also want to thank all these wonderful institutions that have supported me over the years, and I have a company now called Disease that does this microbiome analysis stuff. So if you're interested in that stuff, I'd be happy to help. Thank you. Do you have time for questions or something? Or? Yeah. Okay. Do you have a question? Or? Oh, there's one question over there. Yes.
So the method that you used to reduce the dimension, it was like it's part of the K, right? And then the second question is why did you use that method to reduce the dimension of your data?
So your first question was the method that we used to reduce the dimensionality is K? I didn't quite get it.
Like you went from 10,000 to 100, right?
To 100. All right. So that's not reducing I wouldn't say that's reducing the dimensionality. So the question is, how do we reduce the number of proteins from 10,000 to 100? And what we did was we ran a classic statistical test called the Kolomogorv-Smirnov test, the KS test. So this is kind of like a t-test, but it's non-parametric, right? So it's that you're just doing hypothesis testing. And your second question was? I answered it. Sweet. 2 for 1. Okay. I think seeing you offline. Questions. Great.